Hello, my name is Michelle Morand. I'm a precision cancer medicine educator and advocate, and I am here today with Alexander Rolland, the cancer guy, specialist in precision cancer medicine research. Alex has put together a presentation for anybody out there who might be dealing with urothelial cancer or know somebody who is. There's a new FDA approval for an exciting new drug, and Alex is going to tell us all about that and how it works and how to know if you are a candidate. Alex, take it away.
I'd like to talk about this new FDA approval for
Yeah.
FGFR alterations. FGFR stands for Fibroblast Growth Factor Receptor. In this particular case, we're looking at FGFR3. The FGFR gene is one of a part of family of 4 different genes: FGFR1, 2, 3, and 4. These are receptors, so they're on the outside of a cell. Typically, although not all receptors are on the outside of a cell. Typically, when I talk about receptors, I place them on the outside of a cell, even if they are internalized receptors, just because it helps people understand the process. In this particular case, we're focusing on the FGFR3 gene, and it's unique in that it produces various forms of a protein with different forms found within different tissue types. So depending on the tissue, it's going to produce a slightly different gene product. And this gene, which makes it a good target because this is obviously a mechanism that cancers use. So the FGFR gene is responsible for multiple growth factor interactions. In other words, it's responsible for creating new tissues and so on. It's fibroblasts are cells that are typically part of the immune system that are in the tumor microenvironment, and they can sometimes be recruited against tumors. So.
Okay.
More on that in a different video.
Okay.
So basically, this fibroblast growth factor receptor 3, FGFR3, is triggered to turn on when one of 2 ligands, either FGF8 or FGF9, bind onto it. When one of these binds onto it, you don't need to have 2, just either one. When one of those binds onto the FGFR3, it causes 2 copies of the receptor to come together. This is a common mechanism in receptors, and it's called dimerization. And when that dimerization happens, what happens is it allows one of those receptors to become what we call phosphorylated. That turns it on. Sometimes you can have dual phosphorylation. I believe in this case it's a single phosphorylation, not important. But basically all you need to know is the binding causes a conformational change that causes 2 copies of the receptor to come together. And when they're together, they're turned on. And when they're turned on, basically, this is a normal process that happens whenever you need to replace cells. When you want to mature cells or form new blood vessels, wound healing, bone growth, all kinds of different processes. Depending on which tissue the receptor is in, it will cause growth and development of that particular tissue.
Okay, so far we're talking about a natural, normal, healthy process that happens all the time in your body.
Yeah. Whenever you need to replace tissue.
Right. Okay.
And that's what oncogenes are. They're genes that are involved in replacing tissue, but they're normally turned off until they're needed. So, for example, if you cut yourself, or if you have an injury or a cell dies, then these genes will get turned on. These oncogenes, they'll get turned on briefly just to produce new tissue, and then they'll get turned off.
Okay.
So this is how it works. Here's the mechanism I took from a study on glioblastomas. So this is just a mechanism. As you can see, you have 2 FGFR3 monomers. They come together and they dimerize, and there's a big P, which stands for phosphorylation. And you can see the pathways activated. You can see the top is the cell membrane there. And you can see one of the pathways is the STAT pathway, which results in increased proliferation and survival.
Of the cell or of the person?
This.
Okay, thank you. Just checking. This is not my wheelhouse.
One cell.
Okay. All right.
A great question.
They say there are no stupid questions. I'm just checking in here.
However, in cancer,
thank you.
The FGFR3 gene can be mutated, overexpressed, or it can form a new gene by combining with the gene that is always turned on, which is called a fusion gene. Fusion genes are actually a combination of a gene. So think of a gene as a light switch that is on or off, and then the coding region for the gene attached together. This is called the promoter, and then this is called the actual coding of what the gene produces. So what can happen is you can have this promoter that's either turned on all the time from a different gene that's turned on all the time attached to the FGFR3 gene, which is normally not turned on all the time. And so that causes it to stay turned on. And this is what a fusion gene is it's a combination of two parts of a gene together that causes that gene to have typically oncogenic activity. So since this gene is normally turned off, unless it's needed for healthy cell development, when it gets stuck on, this activity can drive cancer development. Additionally, this particular gene can have alterations in the process where, so when you have a piece of DNA, it gets translated and turned into a final protein as an intermediate step of RNA. And so what happens is the gene gets turned on for a certain period of time, produces a bunch of RNA, and then those RNAs act as templates that amino acids bind onto them, and then you get the final gene product. What is interesting about this particular gene, especially in bladder cancers, is it can have these changes to the RNA. They're called post-translational modifications, and it actually changes the structure of the gene and maybe puts in different amino acids and so on, depending on which tissues it's in. And so just recall that RNA is an intermediate step from when a gene gets translated from DNA into its final form, the FGFR3 protein. So yeah, this is a bit of a unique mechanism where, more opportunity for change. Obviously, this is an evolutionary device that allows this same gene to be used in multiple different tissues. But when it happens in cancer, obviously, that's not such a good thing. FGFR alterations in general are found in metastatic urothelial cancer, non-muscle invasive bladder cancer, cholangiocarcinoma, which is bile duct cancers, non-small cell lung cancers, HCC or hepatocellular carcinomas, brain tumors such as glioblastomas, breast cancers, ovarian cancers, and head and neck cancers. However, mutated FGFR3 is present in up to 37% of upper tract urothelial cancers. And so this makes it a good target. Almost 40% of patients with this kind of cancer are going to have this FGFR3 mutation.
Yeah.
Or have a FGFR3 mutation.
Okay.
January 19th, 2024, FDA-Approved Erdafitinib, which is a pan-FGFR inhibitor. I'll explain exactly what that means. And they approved it for locally advanced or metastatic urothelial carcinoma with FGFR3 alterations, whose disease has progressed on or after at least one line of prior systemic therapy.
Okay.
And this is all based on the THOR trial for metastatic urothelial carcinoma harboring select FGFR3 alterations. In this study, the patients had received 1 to 2 prior systemic treatments, including a PD-1 or PD-L1 inhibitor, so immune therapy. So you can look at the full approvals there.
Right. So probably when they're talking about first line systemic, it's like chemo with perhaps some immune therapy is what they would have had.
Yes, well, usually it's surgery and then obviously chemo and then immune therapy, but that's a different subject. We'll talk about that. Immune therapy is increasingly becoming a great tool in this type of cancer, as are antibody drug conjugates. But this is kind of a targeted therapy that it has a unique position in treatment.
It for the right person.
Exactly.
Yeah.
Right drug at the right time for the right person.
Got it.
So this drug works by binding to inhibiting the enzymatic activity of expressed FGFR1, FGFR2, FGFR3, or FGFR4. So in other words, it is what we call a pan-FGFR inhibitor. So it doesn't just target FGFR3, it targets all 3 of the different partners or the different FGFR genes.
Okay.
So basically, it prevents it from being activated. So I'm just going to go over the THOR trial data. In this particular study, you can see the overall survival, the median overall survival was 12.1 months versus only just under 8 months for the chemo. Progression-free survival, which refers to when 50% of the patients in the trial have their cancer return, was 5.6 months, so almost 6 months in the Erdafitinib group and only 2.7 in the chemo. The thing that I really like to look at, though, is the overall response rate, how many patients out of 100 responded. And as you can see, only 12% responded to chemo, whereas 46% responded to Erdafitinib.
That's
So
why I was asking about that systemic first line.
3 times,
Yeah.
the rate of standard chemo. So, really, I think that speaks a lot to
Mm-hmm.
the power and the potential of
Mm-hmm.
target.
So you have far more likelihood of having another year with this drug than you do with chemo.
Yeah, completely. Yes.
Yeah.
Yeah. I always like to look at the long-term survival curves. Often, as you know, progression-free survival and overall survival are medians. That means when 50% of the patients, it applies to 50% of the patients only. And I like to look at the long-term survival curves because having the medians doesn't always tell you a lot about what's going on. And as you can see up here, the 50% mark, which is the median for overall survival, is right there. And for Erdafitinib is right there. But if you look at this, you can see, the trajectory at, just under 51 months, 50 months or so, 15% of patients are still alive on this. Whereas with chemo, this is like each dot is a patient. You can see, the last patient was just probably about 10, 12% at 39 months.
Three
So
years versus more than 4, essentially.
yeah, so there's a lot of patients with, extended long-term benefits here. And that's why I really like to look at the long-term survival curves.
The other thing I think that's important to note here, you'll probably talk about in a minute too, but just to drop a pin in it, is with thorough sequencing of your tumor, you're going to turn up more than one target, almost certainly.
Yeah,
This is just one mutation. Imagine if you have 2 or 3 that are driving the bus and you're on a combined treatment program that targets all 3. I just want to drop a pin in that for you,
yeah, for sure.
because that's what we're seeing. And that, so this is an obviously excellent benefit to people, but it's not going to be your only option.
Yes. Yeah. And so here's a side effect profile for anyone that needs it. A lot of patients in late stage cancers are going to have side effects regardless. So it does have some side effects.
In contrast to if we go to grade 3, which, those are kind of the less pleasant ones.
That's what we want to look at. Yes.
Yeah. So we've got stomatitis seem to be more present.
Yeah, that's common with EGFR and FGFR inhibitors. Same with dry mouth. Typically, you see that, although we're not seeing it in this particular case.
Okay.
To move on here, the important thing, I think, the real essential thing is that you would never know if this is going to be a treatment option for you if you do not get tumor DNA sequencing. Have to get tumor DNA sequencing. It is the most important thing. It's the basics of precision oncology. Without that, you don't know what drug is going to work for you and what drug is not going to work with you. And I like to emphasize the fact that you need a large panel, not a spot panel. And that is because you also want to know not just the mutations that have targeted drugs for them, but also the mutations that can alter how those targeted drugs work. This is what we call the confounding mutations or the co-mutations. In other words, are there mutations that would prevent this drug from working for you? And, there's lots of data on these sort of things. So really important to get a full panel of tumor DNA sequencing done. I like to do at least 550 genes. There's lots of things to look at there.
It's also, as you were saying about those co-mutations or desensitizing mutations, there are treatments to target those. So it's not just, oh, I'm going to find out that this drug won't work. It's no, I'm going to find out this drug will work, but I also need this.
Have to include another drug. Exactly. Exactly. And so here's a link to a video we did on that. I do recommend everyone watches that video. It's got some important information on the fundamentals of tumor DNA sequencing and why it's important and how to get the information.
The key, obviously, in watching that video is so many folks understand that genetics are important to cancer right now, but they don't know what or how. That video, I think you did a really good job with that presentation, Alex, in talking about all of the elements, and it will help you not get stuck assuming that you've had DNA testing. So many times people call me we have a complimentary consultation process, and I'll be speaking to people and I'll say, oh yes, I've had tumor DNA sequencing, and they haven't. They've had 1 or 2 inherited mutations tested for. or they've had like a 5-gene panel that looked at the most common mutations from 15 years ago, not even present day stuff. So for you to be able to tell for yourself when you've actually had tumor DNA sequencing is very critical, because the faster you get that done and get on the right treatment, the less cancer has a chance to get a foothold. And obviously that's the most important thing here. And
Yes.
making sure what Alex is talking about with the big panel is making sure, as he's saying, that all of your targets are identified, and that any of those mutations that might get in the way of those drugs working are also being targeted. We have a story on the YouTube channel actually right now, somebody by the name of Gail, a wonderful success story that's a perfect example of this, isn't it, Alex?
It is. Yep.
Where
Classic example.
she had a smaller DNA panel and her doctor had put her on a drug for that, but the panel was too small to catch this other mutation that was preventing that drug from working. So just a bit more of a thorough assessment of her tumor has put her into a complete response and great success. Very well worth it. So here we are at the slide where I'm going to tell you, just call Alex if you want some help, really. He and his team will gather all your medical records. They'll look through everything and they'll meet with you for a full hour on Zoom, talk you through everything that needs to be known about your cancer right now, and all possible treatment options and best next steps. You get a recording of the call and a written summary. It's really very comprehensive. We also have a free, this is me, the education and advocacy program. I rope Alex in every week to come and do a live Q&A. Lots of education there for those of you who want to learn about this yourself rather than just have someone else do it for you. Or if funds are an issue, this is a great place to start. And of course, our YouTube channel. Subscribe and stay informed. Thank you, Alex. This was really great. Very helpful information to people who are dealing with that type of cancer, and exciting that there's just another drug that's doing so much better for people.
Yes.
Mhm.
Thanks a lot.
Thank you.
Bye.